Okay, so our last session of this course is a talk on bioethics, and I will just introduce very briefly Dr. Denis Alexander, who is the founding emeritus director of the Faraday Institute, and also an emeritus fellow of St Edmund's College, where we have close academic links just around the corner in Cambridge. He was previously chair of the molecular immunology program and head of the laboratory of lymphocyte signaling and development at the Babraham Institute in Cambridge. At some point before then, he spent 15 years developing university departments and laboratories overseas, especially in Lebanon, in the American University of Beirut. And many of us will have read or heard Dr. Alexander before. He writes, lectures, and broadcasts widely in the field of science and religion on a wide range of topics. And we're looking forward to hear you on key issues for busy church leaders in bioethics. Thank you.
Great, thanks very much, Ruth. And it's wonderful to be here. And I really love coming to these courses because I always learn so many things. Even though you come to a lot of courses, I still learn lots of new things and been learning about AI. And I'm sorry to miss miracles this morning. I gathered you sorted that one out as well. All miracles were arranged. Was that right? Is that what happened? I tell you, okay, a few months ago, I was invited to speak at the Regent's College, which, if you don't know that, is up in Malvern. It's the theological college for the Elim Pentecostal movement, okay, the big center for Elim Pentecostals. You may be some of you here from there, I don't know, and I was asked to speak on are miracles and science compatible? I was really scared, okay, then bringing the scientists into this particular context, but they were very nice people and it was lovely. But I think it's great in these courses, isn't it, that we get challenged and you get our brains sort of amplified just by hearing all these different topics. We go away with sort of ideas buzzing around our heads, and then it takes a while to sort of assimilate and to absorb and to think about it. Thinking about absorbing things, by the way, I don't know what you're like. If you listen to somebody speaking or perhaps in the pulpit, and they come up to the pulpit with, you know, kind of broken leg or their arm in a splint or their forehead is all messed up or something, and you go, I'm a biomedical scientist, so I'm always sitting there and I get distracted. I think, what is wrong with that? I'd like to know, you know, what's going on there, okay? You know, I'm afraid that's the scientist. So just in case there's anybody here in that category. So I lived for 15 years in the Middle East, as Ruth was just mentioning, I think. And so my forehead suffered from sun damage, which was even amplified a little bit last summer. You remember we did have some summer here, if you remember that far back. And anyway, so if you have skin damage, you don't want it to become carcinoma, you don't want it to end up as a melanoma, so you put on this ointment. It's called Efidex. Efidex is actually a chemotherapeutic reagent, if you really want to know, and it will just kill replicating cells. So it mimics the DNA. The DNA gets in with this false bit of genetic letter in there, and it's recognized as DNA damage, boom, and the cells program cell death. And once that happens, okay, then you get inflammation. So that's so my forehead. So now you know. So thank you for one or two people who said, oh, have you had a bike accident? No, not this time, but that's happened also, okay. Anyway, that's just my way of getting us into biology, okay. So bioethics, yeah, where do we begin? I mean, there are so many really serious bioethical issues popping up every day, aren't there? About embryo editing. Should we edit embryos, human embryos, so they turn out to be without their genetic disease? Or should we send our DNA off testing to find out about our ancestry and also about the risk that we have of developing certain diseases in later life? Is that a good idea? Somebody comes in your congregation and wants to know about that. So what would you say? And that's really the sort of things we're going to be thinking about. It's a bottomless pit. There are so many of these things. So what I want to really do is just to give you a sort of roadmap, is to start out by thinking of the standard ethical toolkit that I think is used by pretty much every research council, ethical committee, or ethics committees sitting all around this city as we speak, sorting out whether certain experiments should be done. And then I want to take 3 examples. We have time, we'll take 3 examples, if not 2, of just working through the toolkit, the use of this ethical toolkit to see how it can be used in different contexts with different challenges. Okay, so that's what we're going to do. You're probably very familiar with all these tools already, so I'm going to zip through pretty fast. But just so we're also starting on the same page of what the toolkit comprises. So natural law ethics, ethical toolkit number one, a familiar idea. So the roots of this approach go way back to Aristotle, very much amplified and thought about by Aquinas in the 13th century. According to Aquinas, natural law is one aspect of divine providence. So it's a way of thinking about the natural order, the way that ethical values and so forth are embedded in what we are, in who we are, in the way things are set up. And so the virtuous life is one lived according to nature, fulfilling our natural potential. There are whole books on this, okay, so we're really zipping through quickly here. I always think of it, because I'm a scientist, I'm not a professional bioethicist, I kind of think of it as the yuck factor, okay? If you hear something being done, you might hear one or two in a moment, just to warn you of things that are being done, you might go, ugh, yuck, And in a sense, that reflects a natural law ethics. It's sort of where we feel this is against the natural order. There's something deeply, profoundly wrong here. Second, very different one here is utilitarianism, the second tool in our toolbox. Very much associated with the English philosopher Jeremy Bentham back in the early part of the 19th century, especially, very influential at that time. So what actions will produce the greatest good for the greatest number of people? Who will benefit? Who will not benefit? Are the costs worth it? All the kind of things that governments tend to focus on, when they're thinking about AI or thinking about bioethics and so forth. What will be the consequences of this particular action? So consequentialism. I put utilitarianism and consequentialism together here. They're basically first cousins. They're very close to each other, and they end up in the same place a lot of the time. So very powerful in terms of ethical considerations on ethics committees, very closely connected also to what is sometimes called divine command theory, So, of course, that refers to the claim that morality is ultimately based on commands or character of God, and the morally right action is the one that God therefore commands us to do, something obviously very familiar to us as Christians. And that clearly then leads on to also, sorry, I'm skipping over here. I've jumped a page by mistake there, and I've jumped over to deontological ethics, which is closely the first cousin of divine command theory. So rewind, okay, and go now to number 3, which is deontological ethics. The term is derived from the Greek deon, meaning obligation or duty. So there we've got rule-based ethical systems. We should be doing the right thing, whatever the consequences. We should stop at red traffic lights because that is the right thing to do. If we don't stop at red traffic lights, what kind of influence is that going to have on other people as well as also upon ourselves? I mention that, by the way, because for 5 years in Lebanon, I never stopped at red traffic lights. It's a confession time. In the middle of a civil war, when people are fighting each other, they don't stop at red traffic lights, okay, but they still work, flicking away, there they are. So there are places in the world where you don't stop at red traffic lights, but on the whole, in this country, as I learned when I got back, it's a good thing to do. Okay, deontological, it's a good thing to do. So, and this, now to catch up with what I made a mistake in saying it just a moment ago, this is the one that's closely related clearly to divine command theory, because here God is giving us a command to do something. It's deontological. We have to do it, whatever the consequences might be, that doesn't matter. It's deontological. You just do it. Okay, so that's pretty clear. Fourthly, in our toolkit, we have virtue ethics. What actions will encourage virtuous qualities in myself and in others? If I don't keep stopping at red traffic lights, what bad influence might they have on other people? Other people will see me and they'll go through the red traffic light as well. And so we have an influence in everything that we do. No person is an island, as often is said. We all have influences on other people in what we do and what we don't do and so forth. Now, of course, the problem here sometimes is knowing ahead of time really what a virtuous behavior is in the sense of what influences are going to have on our own lives and those of others. In a sense, in talking about AI, we've just been thinking about that. You know, what are the bad influences it might have? In a sense, we've been doing an exercise in virtue ethics and thinking about AI and its influences and so forth. Right, so those are the main tools in our toolbox. And what I want to do now is simply take 3 issues. We're going to use most of the time on this first one, simply as examples of how do we go about the process of using those tools, as it were, to think about what to do, what we should do, what we ought to do. And of course, ethics is all about what we ought to be doing, what we ought not to be doing. So let's start then with the ethics of human embryo editing, sort of a hot topic. And you might feel, well, this sounds like something really science fiction, something that's not really part of our daily lives in my congregation, not really concerned about that, and so forth. And in a sense, you're right, but it's happening, it's beginning to happen. And also, it will be happening more and more for the next few decades. And it will be something that the young people, certainly in our churches, will be needing to face up to in the next few decades as well. So it's good to be there and know a little bit about it and be thinking about it. But mostly I want to use it as a kind of a work tool, as a test case, if you like, for how to think about these bioethical tools. So what is it? Well, embryo editing has arisen from this huge wave of new technology to modify genes, which is called CRISPR-Cas. It's incredibly widespread now all around the world. It's a bit of genetic engineering technology. And CRISPR-Cas simply refers to a bit of molecular machinery. It doesn't matter if you don't understand all that up on the screen. But simply what it does is it takes this machinery, it takes a pair of scissors, molecular scissors, we call them nucleases, which will get into the cell and which will be targeted to a specific sequence of the DNA letters in the DNA. It does that using an RNA guide that guides it to the right place. Scissors cut, and then it's repaired in a certain way so that you can edit DNA, basically. You can take one single genetic letter out and you can put another one in its place, or you can restore that genetic letter to the one that it is in everybody else, in case that's a genetic letter which is a bad one, which leads to having a genetic disease or something like that. And so this whole technique has really swept across the biological world over the past couple of decades. I just looked up yesterday how many papers came out in the scientific literature just last year, year 2022, which have CRISPR-Cas, or CRISPR actually, in the title, and there it is, it's 2,624 papers, okay? So if you're a scientist trying to keep up with what's going on, you're gonna have a hard time, okay? There's a lot of publications out there. That simply reflects this huge explosion of applications and developments and advances in this particular field. And the use of CRISPR-Cas became particularly famous back in November 2018, when Dr. He Yankui from Shenzhen in China announced, made a dramatic claim, for the first time ever, he had, using CRISPR, he had edited an embryo, a couple of embryos of twins in such a way that he thought they would become resistant to HIV infection. And the reason for doing that was because the parents, or one of the parents, had HIV. And so he thought this would be a very novel new way to try and kind of give protection to these particular children who have indeed been born. And what he did was to delete a particular gene. It doesn't matter again about the details. It's called CCR5. He deleted that gene out of these human embryos of these twins using the CRISPR-Cas technology. And at least one of them, I don't know about the other one, but is reported to have a complete absence of this particular gene. The reason for knocking out the gene was simply this particular gene encodes a protein, CCR5, which is necessary for HIV infection. So in a way, it's not healing, is it? This is actually enhancement. You're actually trying to give protection to those twins against HIV infection. And this created a huge outcry for all kinds of different reasons in the scientific community and generally in publicity. Partly the risks are not really known or well understood at this stage. What happens if CCR5 is needed for normal health? Actually, that probably is the case. And you're knocking out something that you need for other things as well. So you have to be really careful. There are just other ways of preventing HIV infection during a birth. So, you don't really need this anyway. In any case, if you're really going to pioneer a new technology like that and apply it to human embryos, why not choose a terrible, lethal disease and all that kind of thing? Wouldn't that be a lot more sensible? So there's all kinds of criticisms made, and then it turned out that he hadn't even passed it through the local ethics committee in Shenzhen University, and so he was sent to prison anyway, and he was just released a little bit towards the end of last year, actually, 2022. And there are many other attempts being made to use CRISPR-Cas for healing human embryos. Okay, that's going on as we speak. So it's good to remember there are about 7,000 human genetic diseases. It depends, you'll see numbers which vary a bit, it depends what you count in there. That's including various subtypes of different diseases. But these are caused by a single mutation, single change, variation in a single gene genetic letter. One single letter, okay, is changed. That's enough to cause some pretty nasty genetic diseases. Some of them cause the deaths of kids in the very early years. That's, by the way, why I ended up in Lebanon to help set up the National Unit of Human Genetics many years ago now, because a lot of Lebanese marry their first cousin. Not a good idea if you want to avoid genetic diseases. So this embryo editing it's a research enterprise. It's not allowed. You can't actually do it in a way that will lead to the birth of embryos in any country of the world, either by specific legislation or by ethics committees and so on. Okay, so, but it's coming, it'll come, and it'll continue to be a big issue, I think, in the next 10, 20 years. So the main question anyway we have here is, how do we apply our ethical toolkit to assess human embryo editing? What do we do? Let's work through the different tools, see how they might be able to help us or not in this particular discussion. So natural law ethics, it might say, well, what is the technique doing? If you're trying to heal a human embryo, you're just taking the abnormal genetic letter and you're taking it back to normality. You're taking it back to what it should be. That will give you a healthy child when the child is born, at least free from that particular disease. And so I think natural law ethics could be quite favorable to this technology for that reason, okay? Because you're restoring the natural order, maybe. Utilitarianism, well, the outcome of the procedure sounds pretty good, isn't it? Because after all, what you're doing is you're healing an embryo. Here's this genetic disease that's been running through a whole family for many generations. You want to get rid of it. Why? If you've got a technology that would lead to the birth of children that wouldn't have that horrible disease, isn't that a good thing? So I think utilitarianism might be pretty positive about it. Although at the same time, I think many utilitarians would want to say, well, watch out the slippery slope ahead, okay? Because how will this technology, if we develop this technology now, if we perfect it, how is it going to be used in the future? Is this going to be used, to try and design babies so they'll get their blue eyes or their long hair or their short hair or their whatever, trivial things? Is it going to be used for that? It's going to be used for enhancement. And so there are all kinds of issues raised as you kind of develop a technology in this way. Technology is never neutral, not really. Now, in one sense it is, as we've been hearing, you can use it for good or evil. And in another sense it isn't, because it gives you a potential to do things you couldn't do before. So it's loaded, okay? So technology is not neutral in one sense, because it's loaded with potentiality that then becomes available. And we see it in AI, don't we? We see it in AI, you do all kinds of things, wonderful. And so that one thing leads to another. Utilitarianism might point out that in any case, let's say 99 cases out of 100, there's no point anyway in genetically healing an embryo because you've got pre-implantation diagnosis. That's a much simpler method. So what is that? Okay, in pre-implantation diagnosis, you take an embryo, you have to have in vitro fertilization, you have IVF, and you wait until the embryo in the lab is about 8 cells, and you can just pick out 1 cell, and then you can screen the embryos in the lab to see which embryos do or do not carry a particular genetic disease. This is where you've got a family history of genetic disease. You know what might come in that family, and you know what's possible. You looked at the parents, you know that both parents are carriers, and so you can screen the embryos and just with 1 single cell and do DNA analysis and show that embryo will not be a carrier of that disease. Right, pretty amazing actually. On the NHS, what is it now? A couple of 100 diseases you can screen. I don't know, something like that. It's going up all the time, so my number there might be a bit out of date. Now, there's one exception, one kind of exception I put up there, where there's certain cases where, very rare cases, you've got both parents who are carriers of what we call a dominant gene. That is the gene itself, just 1 copy of the gene out of the 2 copies. Remember, there are 2 copies of each gene, thankfully we have. And so 1 copy will cause, let's say, Huntington's disease. And if both parents are carriers, then all the embryos will, if they're implanted, will then lead to offspring that will have Huntington's disease later on in life, much later. Okay. Well, that's a very rare example. Okay. Are we really saying it's worth developing that whole technology for that? There's very, very rare examples. Well, you know, that's a point for discussion. Also, we might want to move to the deontological perspective. Is it right? Is it right to manipulate embryos in any way? Remembering, of course, that if you do in vitro fertilization of this kind, there will be wastage of embryos. Okay, you're going to throw some away, right? Okay, and so that then raises, of course, this real thorny issue of what is the status of the early human embryo. And there are 3 main beliefs you'll find anywhere in the world. You go around anywhere in the world, and these are the 3 you'll find, and they'll be here in this room. 2 of them, I think, will be here in the room. I don't think the 3rd one will be here. I hope not. Anyway, so belief A, the zygote, the fertilized egg, is a human person, and it has all the rights of personhood. Very common belief in the Christian community, right? Belief B, a slight modification here. The zygote, this fertilized egg, this very early embryo, it's a potential person, and therefore deserves our protection, but not at any cost. Not at any cost. And then belief C, the kind of materialist view, well, you know, it's just a little bunch of cells, it's smaller than a pinhead, throw it away, it doesn't really matter. Okay, that's kind of a more materialist view. So that also, obviously, depending where you end up on that, I'm not going to say what you should believe, what you shouldn't, I'm just raising the questions, that's the easy part, okay? But that's something to think about. What about virtue ethics? Well, I mean, this position might suggest that the goal to achieve perfect health is anyway an illusion, and that trying to eliminate a human disease in the population somehow diminishes the value of the other people in that population who suffer from that particular disease or disability or whatever it might be. Discuss again. And virtue ethics, I think, might also point out that showing love within the family does not necessarily entail having genetically related children. So a better option might be adoption. It might be having a baby by in vitro fertilization, possibly, and so forth, where that is possible with using healthy donor sperm or something like that. There are other possibilities that one can think about to avoid embryo editing if we really feel that is not the way to go. And I think virtue ethics is of particular relevance when thinking about genetic enhancement. It's already been sort of highlighted, I think, previously in this workshop that there's a big difference really here, isn't there, between healing and enhancement. So Transhumanists, as we well know, would like to create people with advanced intelligence or with athletic abilities, super health, all that kind of thing. This is the transhumanist push that's pretty powerful, actually, especially powerful in Silicon Valley, and is in the cultural air we breathe through films and movies and books and so on. I think one thing to think about in that context is, in a way, the complexity of the genome. The genome is simply all the DNA, that information that we have in our bodies. That complexity is its own protection in some ways. If you just think about intelligence, yeah, intelligence has some genetic contributions to it, but the contribution of any one genetic variant to the variation in intelligence in the population is certainly much less than 0.1%. Okay, 0.1%. And there are probably thousands of genetic variants that are involved in contributions of genetics to intelligence. There are contributions, they are there, but there are thousands of these variants, okay, involved in early development and so forth. And so are we going to change thousands of genes in an embryo? No, I don't think that's on the horizon, even in science fiction scenarios. So really, that's something to think about. So in the way, the genome has its own complexity, which is its own protection and so forth. And apart from that, you know, would we really want to change trivial things in a genome, as I say? And one way of looking at it, I like this picture from Michelangelo's creation of the sun, moon, and vegetation. This is the Siena Sistine Chapel in Rome, and painted between 1508 and 1512, by the way. And it's really amazing. If you've been to, probably many here have been to Sistine Chapel and just stared up in awe really at this amazing painting. It was actually restored over the period 1980 to 1994. Huge restoration. And you can see the pre-restoration at the top there, and then the post-restoration, I hope you can see it is different. Okay. Of course, the restorers, their main aim is to restore that beautiful Michelangelo's paintings to the original, what he had in his mind, to restore it to original colors as much as possible. Not to change it, not to enhance it. The restorer of a painting is not to enhance what the creator has brought into being. I think that helps us, doesn't it, in our thinking about the difference between healing and enhancement in terms of bioethics and these latest possibilities that we have to meddle around with embryos and so forth. But if we come back to the immediate aim of human embryo editing, which is the healing aspect, you know, we're kind of left with this question, aren't we? You know, would Jesus heal embryos? Just to put it a bit bluntly, you know, is that something that Jesus would do? Or if you like, would Jesus delegate that now to us as healers to do that, medical healers or in other ways? Is that what we're supposed to be doing? I think it's good to go back to Genesis 1. We often do that. We did it a bit yesterday, but let's go back in just to think briefly about being made in the image of God, humankind. Very familiar with the passage, Genesis 1:26. We touched on it yesterday. I think it was Matthew talking about the various interpretations of what it means to be made in the image of God. The Adam, the humankind, male and female, he made them and so forth. I often think, how would the first readers of Genesis have really understood this? And as you probably know, the image of God language at the time, when Genesis was being written and edited and so forth, it referred to kings and priests in the ancient languages of Egypt and Mesopotamia. So it's kingly, priestly languages being used here. And then suddenly Genesis 1 goes, hey, the whole of humankind are kings and priests. It's very revolutionary. You know, Genesis 1 is very political. Wow, if you think of the context in which it was written, there's a powerful political revolution going on there, because instead of having these, you know, these people who's in charge of the serfs slaving away in the fields and saying, hey, humankind are kings and priests. Very powerful indeed, I think. And I think often we miss the sort of political power and revolutionary power of Genesis 1 when we read it. People think it's old text, you know. In the context, it was absolutely revolutionary. Actually, turned the world upside down, and has been turning the world upside down ever since. And once we get that, I think, then we start thinking about, well, what does that mean in terms of our daily lives and so forth? Well, it certainly means that every human individual has a status and a value which is irrespective of their education, of their genetic status, of whatever it might be, and so forth. And so those kingly and priestly roles are now poured out on the whole of humankind. And of course, some people, some of us have a greater ability to practice those roles more than others. Others are recipients of those roles. We are recipients of care. We're equally part of the image of God as a recipient of care or as a giver of care. Yeah, it's a very key point, actually. We're all part, humankind, a community. Often, image of God language is used, being Westerners, used to me as my individual. It's given to Adam, humankind, the whole of humankind are made in the image of God. So it's a community theology. And I think that is reflected then in medical care and all kinds of other ways as well. So coming back then to embryo editing, well, you could argue that acts as a powerful driver to use this technology. We're going to subdue the earth. Remember, we're just being reminded of that yesterday again from Matthew. We want to subdue the earth. Part of subduing, I think cancer research is subduing the earth. I spent a long time in cancer research, subduing the earth. And so genetic engineering, yeah, it's great. You can use it for subduing the earth. That's wonderful. So how far do we go? Okay, then how far do we go? And then, of course, we think about the possible misuses of it, which could be quite trivial. As I said before, is it going to be used for people who want to have children, with blue eyes and certain colored hair, or I don't know, who are good sports people or that kind of thing? Probably not, no. And what about the risks as well? Thinking about risks of future disease. We all have a 1 in 2 risk in this country, our population. It's about a 1 in 2 risk of having cancer now. Any time during our life, the whole of our lifetime risk is 1 out of 2, 50%. It's pretty high, isn't it? Okay, so are we going to delete half the population in order to avoid half the population getting cancer? I don't think that's going to happen, okay? So, so we have to think in those terms as well. And then virtue ethics. What kind of society are we creating if we aim to eliminate all kinds of genetic diversity that might be possibly be deleterious? Okay, so that's another thing to think about. And then, of course, we have the consequentialist worry about the slippery slope and exactly where this technology is going to be going. So I think also in thinking about, as we baptize our toolkit into Christian theology, we also have to think about wastage of embryos and so forth. And for some people who have belief A, they really believe that early embryo has the full, not just potential, but has the full rights, fully a human person, then clearly IVF is out. It's out. It has to be. Because, if you believe that, then you won't want to deliberately do any kind of technology that will lead to embryo wastage. Okay, so that where you start from in this discussion will end you up in slightly different places and so on. I should mention, by the way, there's a new Faraday paper coming out soon by Keith Fox, which is about ethical issues in human genetic engineering. It's not out yet. It should be out very soon, very soon. Great. I'm going to spend a bit more time now on a second application, which is something nearer to home, nearer to our own congregation, something that maybe some of us have done here. And that's the ethics of direct-to-consumer testing. So what about sending off your, saliva, spittle, boom, spit it off. A lot of spitting there, send it off, and you'll learn about your ancestry, what fun. And in many cases, if you pay a little extra, you can also get a whole range of risk factors that you might develop some nasty thing in future life. Okay. Hey, well, it's £79. And this is what's this one, 23andMe, this one is, there are dozens of the gene testing websites now. In fact, 23andMe sold over 250,000 genomic test kits in the UK as of June 2020. In that particular year, they've gone up from 190,000 the year before. I mean, it's big business, big business. I think it's something like 100 million Americans now have had their genes tested and so on. Okay. And you never know quite sure where that DNA information gets to. Okay, there's some examples of that. And so there you are. You don't have a counselor handy, or you've got the nice printout at home, and it tells you all kinds of information that you didn't know before. And as I say, I mean, some of the the way it's sold is to be responsible about your health. You see, you should be more responsible about your health. So you get this information so that you know, now I know how to behave. I should be more careful because I have a high risk of developing heart disease or something like that. Right, okay. Now let's just go through quickly then the toolkit. So natural ethics, I don't think it's got much to say about this. There are certain movements or groups who believe DNA is sort of sacred somehow. You shouldn't touch DNA in any kind of way. You shouldn't even find out about it. I don't think, that's certainly not a Christian view, I would say, but maybe some people have that kind of view. Deontological ethics, well, it's keeping to the regulations. This whole testing procedures in this country are regulation free at the moment. Pretty much you do what you want, okay? It's a bit worrying, actually. They need to be regulated, I think, because of the implications, which we'll come to in a moment. Because as soon as you get to utilitarian consequentialist ethics, then you suddenly have a minefield. Okay, a whole lot of stuff comes out there. And just to give a little background information on this, what we use here, what's used in these technologies is called SNP chip genotyping. And that's what it is. It means you can screen maybe 700,000 different variants just in one little chip. So the SNP stands for SNP, it's single nucleotide polymorphism. This might be worth knowing, that little acronym actually is useful language. So polymorphism is just a technical term meaning a variant genetic letter in that particular place in the DNA. Nucleotide is a chemical name for a genetic letter. It's all very simple. Okay, so SNPs. And so all of us have around about 5 million SNPs in our bodies right now as we sit here, which are, in other words, these are variants which are different from us and the person next door to us. Okay, it's actually between 4 and 5 million, something like that. So they'll look at about 700,000 of these, and based on that, they'll give you a nice ancestry readout. They'll tell you that you are 25% Swedish, and you're 13% Italian, and whatever, you know, they give you what seems to be very, you know, just fun stuff. What people don't realize is this. Now we're just thinking about the ancestry side of things. There are consequences, okay. One is a lot of people who've done tests have found out their father is not their real father. We're thinking about that. A lot of people have found and they will tell you often, some, not all the companies, some of them will tell you if you've got relatives you know, you've got an aunt, an uncle, you've got cousins living in some other country, you didn't know anything about them. Ooh, what does that mean? Who are they? I'm related to them, okay? And suddenly you get a flood of information that people didn't know they were going to get, really. They didn't realize the implications there. And so there can be some pretty bad consequences. And sometimes the tests have led to divorce, family breakdown, because there's things that might have been best never to be known, okay? Information is powerful. Information is powerful, okay? And I'm going to go a little beyond because I started at 3 minutes past, so just looking at my good time thinking, okay. What about medical reasons? Medical reasons, and sometimes people don't also realize the full implications of these as well. So the SNP chips, if they detect, sometimes they're very rare variants. You could be, in your genome, you might have a variant no one else has got in the whole world. You don't know, probably a good thing, doesn't matter. But sometimes these SNP chips will detect one of those. There are nearly always false positives, actually. You can detect them much better by whole genome sequencing, but usually with the technology, they are false positives. And so therefore, you might be told that you are at risk of having some terrible disease. Oh, in 30 years' time, oh, you'll live with that. It's wrong. You know, there's a lot of that around, actually, and it's just wrong. It might be right, but anyway, so that's a problem. And there are also false negatives as well, quite often false negatives. So it didn't pick up because there are many different variants that can be predictors for the same disease, you see. So the chip might only have one of those variants in there and doesn't have the other ones, okay. So therefore you might be led into say, oh, I don't have a high risk of heart disease, so I can relax, okay, and live it up, you know, and have a lot of McDonald's. No, I didn't say that. A lot of food from not the healthiest places, and so on. And so, and that's dangerous, okay. So And actually, the reality is that in most cases, we can't do anything about it anyway. You know, you have some risk factor. You know, if we just have our 5 veg a day, if we have daily exercise, try not to put on too much weight, you know, that's bottom line what you need to do. Okay. And as I said, we already have this 1 in 2 risk of anyway having cancer and so forth. So what about virtue ethics? Well, ironically, I mean, the thing is that it can actually lead to unnecessary disruption of family life. As I've already mentioned, it can lead to worries and concerns, and, you know, which really needn't be there. Okay, so that can be something to think about. And also, possibly virtue ethics in reverse of that might suggest people should be thankful for the genes that God has given to them as a unique gift for their particular lives and personalities. And we should be thankful for the variant genes that God has given us as part of our own identity, as part of our own human genome, and we should give thanks to the Lord for that. We could talk a lot more about that. As I said, I thought I'd run out of time for the third example, which we won't go into, which is, by the way, about monkey-human hybrid embryos, which you probably don't want to hear about just before tea or just after tea anyway. But anyway, so I'll flag up this book, which does give you a little introduction to some of the genetics, and that's on the bookstall here. Yeah, it's much reduced, so that's a good thing. But if you're interested in the genetics, there's a chapter there giving some introduction to that. But also that's more about behavioral genetics and how that sometimes can be misinterpreted by people as well. But I'll leave it at that, and we'll just have some time for discussion and chat. Thank you very much.
Thank you very much. So our online participants will join us, and we'll have questions either from in here or online. Anyone want to kick us off with a yes, Devinder?
What would the toolkit have to say about monkey-human hybrid embryos?
Okay. All right. Oh, well. Okay. So I can show you that was the news report came out last year, which has had a lot of publicity in the newspapers about the human animal cell chimera, an embryo which is formed. And you do this by taking human stem cells, which are the type of cells that can develop into anything in our bodies. We all start out as stem cells, and then they end up becoming kidney cells or skin cells or brain cells or whatever. Before they go in that route, they'll start out as stem cells. So what the experimenters did in California, by the way, they took some human stem cells and they injected them into a macaque monkey embryo. And the human stem cells then start multiplying. And even in that monkey environment, they can grow. And after 20 days, 4% of the embryo was human cells, right? So, and why did they do that? Well, they did that because they're interested in early human development and abnormalities that happen in human development. And this is one way they want to look at the way that early human cells develop in an embryo in that context. So that's their justification, right or wrong, but that's why it was done. And so you can sort of see where that might go. Okay, now clearly it's entirely forbidden to have births of embryos of that kind, absolutely outlawed and so forth. They would go to jail if they did that, no doubt about it. It's America, after all, you go to jail, okay? But, you know, so it raises questions, doesn't it? You know, I suppose for the future, if you want to be science fiction, then it raises questions for the future about what status does that embryo have. It's one day, some crazy scientist somewhere in the world, we've seen already from the one in China, it can happen. You know, you can have a crazy scientist who does something which is entirely out of the normal ethical framework accepted by the scientific community all around the world. And so we have to think about it, you know, what is allowable, what isn't allowable. And to put it crudely, I mean, being made in the image of God, what percentage of human cells, well, let's put it this way, if you had, what percentage of monkey cells would you need mixed with the human cells until you're no longer made in the image of God? Well, okay, that could be a discussion that would keep us going for a while, wouldn't it? But we have to just sort of be aware of some of this stuff's going on, and we quite likely actually will be asked questions about it. People in our congregations, they look at the media, they look at what's going on, and they go, "Whoa, what's happening here?" And they might come to their pastor, the church leader, and say, "What do you think?" And so it's good to have some sort of ideas about that, I think. So we're a little bit ready. We can't be expert on everything, but at least to have some sort of thoughts in our minds about that. Yeah.
Thank you.
Yes, are there any questions online? We've got several here. We'll give you time to raise your hands online, but we'll go first to another one in.
It's probably a silly question, but when you're editing an embryo, is it just when it's a single cell, or is it like multiple cells? And do you have to edit every single cell that's there? And if that's the case, then can it be done to adults?
Right, no, it's a very good question, thanks. Yeah, so the idea is in the very early embryo would be a blastocyst, which is about 8 cells, okay? And so the technology aims to repair every single one of those cells. Now, what hasn't been published, the only time it's been done is the Chinese experiment that I mentioned, done back a few years ago now. And one of the questions raised was, did they test, is that child that's born what we call a chimera? In other words, are there cells possibly within that child who has been born that weren't where the correction wasn't made in that very early embryo? The aim, though, the whole purpose of it is to edit the embryo, meaning all of those 8 cells at that point, or even earlier than that. Okay, it's early enough so that then all the other cells that come out will carry on having that corrected genome, the corrected DNA in there. That's the aim of the whole thing. But now you say, could we do that in the adult? Well, we wouldn't be able to correct all the cells in the adult, no. I mean, we've got 10 to the 13 cells, okay, something like that in our bodies, okay, so a huge number. So then genetic engineering goes in a different direction, which may be to try and, it depends what the disease is, but sickle cell anemia, has there been recently cases where genetic engineering has cured the sickle cell anemia? That's just in the past year, there have been publications on that, which is really exciting actually, but that's not in the germline. That is using another technology to get cells into the body that will grow in the bone marrow and will then produce the missing protein, whatever it is. Okay.
Can you say what the germline is?
Sorry, the germline is the cells that are passed on from a generation to generation. So sperm, eggs are all about the germline, what we're passing on to the next generation. So the germ cells are in that category, but the somatic cells, all that make up our bodies, are not. The information here is simply for me in this generation, in my body for this life, you see. So when we're thinking about genetic engineering, we have to think about the difference between the somatic cells and the germline cells, and that's very, very different, very different. And of course, once you start messing about with the germline cells, another question is, well, the child that is to be born, they didn't give permission for their bodies to be manipulated in this kind of way, you see. So that's another question that can come up. Now, if they're cured from sickle cell anemia or something, it's not a nasty disease to have. I think most children would be very happy that when they realize perhaps in later life, they were born without that particular disease. I think they'll go, hallelujah, when they look at people who've got sickle cell anemia and then realize that, I'm going back to embryo editing here, maybe they were cured, at an early stage or something, then they might be very happy about that. So we have to sort of think about that too. Thank you.
Great. I don't think we have any more questions online just now, so we'll go to Shami.
Yeah, thank you so much. So my question is there a correlation between having trauma in our DNA, so as a result of slavery or as a result of the Holocaust, that would then affect our mental health generations down the line. So is there any correlation? And if there is there anything that science can do, or is that a permanent indentation within our DNA that would then give us a particular predisposition given our ancestral background?
Thank you. That's a great question, and one which is taking a lot of people, you know, there are labs working on that, definitely. And so there are some classic examples which you'll find, probably actually you'll find in that book, I suspect I put them there, but, or maybe it's other books, I can't remember. But anyway, there are classic examples. One of them is the, what's called the Dutch winter of 1940-41, where the Nazis surrounded Holland, the Netherlands, and a lot of people were starving. And so, so what they looked at was the pregnant women at that time. They've got the records. You see, you need a country where you've got good records of what's going on. And so they looked at the progeny of the women who were starving at that time and what happened to them, and not only to them, but also to their grandchildren as well. Now, this is all to do with epigenetics. So epigenetics is, what that means is the modification of DNA from environmental influences of some kind. And that's going on in our bodies all the time as we sit here. So when you had your tea, you changed your epigenome. Okay, in other words, you started having modifications of bits of your DNA in such a way that they regulate secretion of digestive hormones, that you would digest your cake properly. Okay, so that's all to do with epigenetics. But epigenetics can be inherited. In other words, things that happen in my life to my epigenome can be passed on to my progeny. That is possible. Where it gets tricky then is, okay, so now let's go back to our Dutch starving pregnant mothers in 1941. Okay, so was the starvation, of course, bad nutrition in pregnancy is bad for anybody. It's bad for the offspring anyway. We all know that. Okay, so to what degree is the offspring suffering from something because of an epigenetic change, or was it just simply because of their growth? They didn't grow at the wrong, you know, they didn't have all the nutrition they needed at a certain point of fetal development. It gets a bit complicated. But there's quite good evidence in the Dutch case that the third generation, that, you know, after the mother in the 1941 winter of starvation, then had children who did have some problems, especially type 2 diabetes, things like that. And then their children, there's some evidence their grandchildren have some medical problem there. The other famous example is from Sweden. Similar sort of thing, goes way back to the late 19th century. Very good parish records. You can track again, the harvest time was either abundant and wonderful, or it was really bad. Okay, it went up and down, up and down. And that's been correlated with the health of the grandchildren in a similar way. So those are two major examples which have fairly good data. Now, but to come really to the heart of your question, I mean, these are things like, as I say, type 2 diabetes, instance, and so forth, that sort of thing. That's very different from a mental thing where you have some, you know, sort of deep sense of trauma in the grandparents or something. There's no, as far as I know, there's no good evidence for that. Okay, that kind of experience. One thing where it gets very tricky is if your grandparents were in the Holocaust, you know, then the environmental influence is huge from that experience. Okay, you were brought up as somehow you were the only one, your parents survived, you know, and the others all got killed. You know that from your family history. That's a powerful influence in your environmental upbringing and thought, isn't it? So, yeah, so therefore it gets tricky, you know, again, genes, environment, you know, how to bring that sort of together to really understand that. But I don't think there's any epigenetic data in humans anyway. There are in animals, but not in humans for some sort of third generation experience and what happened to the grandparents in some way, as far as I know. But that's a great question, and it is a fascinating one. Yeah.
I was just going to.
Yeah.
It was just the reason why I was asking as well was coming from Zimbabwe. We know, like with instance with the elephants, if you killed a a cup of an elephant, and then probably a couple of years down the line, the family remembers who was responsible for killing, and so it will actually attack the individual. So this is from folklore. So whether it's been proven yet, I'd have to definitely, you know, ask my grandparents, but it was stories that was always passed to us that never injure an elephant, because it then passes on that information down to the other elephants that will come, so that if that person then injures, or the elephants remember, they will go and attack that person if the person was in that particular vicinity. So it's just to check whether, yeah.
That's amazing. Thank you for that. Thank you. Yeah.
Could I ask a question?
Great. I'm just checking whether there's anyone online who would like to ask a question. Do feel free. And then I think not. I don't see any. Oh, Felicity. Great. Well, let's have someone from online before we go back to the room. Great. Thank you.
I am not a biologist, but I believe it is accepted as true that we have many, many cells of other living organisms inside our bodies. I have this vague recollection that they actually outnumber our own cells, but I might be wrong about that, without which we couldn't survive. So you made some comment in the case of a chimera, a human being that has cells within them of different DNA than the majority, or has the cells of two different kinds of DNA. Can this be compared to our microbiome, our collection of microorganisms within us, which don't share our DNA and presumably won't be treated the same way as us, aren't considered as equal with us as individual organisms with rights?
Lovely. No, that's a great question. Thanks very much indeed for that. Yeah. So it's about, you know, it's called the microbiome. So all the bugs in our guts. Sorry to talk about this just before you're having your dinner, but you know, this is biology. Anyway, so, you know, about 10% of our bodies are bugs, basically. It's something like, don't quote me on that. The ratio has been changing a bit recently, actually, that people have been playing around with the numbers. So, but anyway, a very large proportion of our bodies are bugs. Okay, that's a microbiome, especially in our intestines. You know, they are very rich in a very large number of bugs, bacteria. Okay, which rightly, as Felicity is saying, have different genomes. They have different DNA. Okay, so are we then a chimera in that sense? In one sense, we are. I mean, it depends on how you define chimera, really. But generally speaking, in biology, generally speaking, the word chimera is kept for an actual body, so that, for example, you know, here I would have cells from an animal down here in my palm or something. I'd have cells from a monkey. Over here, those would be human, you know, and all that sort of things. In other words, integrated thoroughly into our bodies is the normal way of understanding a chimera in biology. But in a sense, you're right. I mean, all those wonderful bugs, I think it's all part of our identity, you see. I could go on a long time about bugs, I better not, but that microbiome, those bacteria are really important for our immune systems. You know, really important for our immune systems. And to have a good, healthy immune system is linked to the type of bacteria we have and so on. There's a lot of pharmaceutical companies right now doing research on the relationship between the bugs in your guts and your health in terms of mental health as well, which is quite fascinating, isn't it? To think that bugs down here might have some effect on our mental health. Well, I don't know if you find that an encouraging thought or not encouraging, but anyway, it's certainly a lot of research in that area.
Let's have one more. Any delegates with any burning questions before we? Yes, Jonathan.
Just one little question, Doctor. Thanks. Would it be okay to eat a steak that's made in a laboratory?
Yeah, I would have no problem at all. But it depends on the laboratory and depends how well they've done the steak. You know, I mean, some of these synthetic foods which are being made, those are really important for the future of humanity on this planet, because, you know, we're running out of all kinds of things. So, but I think in principle, I have nothing personally against that in principle, because it's just good food, you know. I mean, if you think about all the synthetic things we eat every day, it's unbelievable. You know, the list goes on and on. Okay, and so I personally would have no problem about that in principle. I would just like to know what the taste is and what sauce to have with it, basically. I know that would be more my concern. Okay, but I wouldn't have anything against that simply because it's been made in the lab. I mean, what, you know, that's my view, since God gives all these good gifts, you know, and God gives gifts through people, and God gives through good chefs and good making, you know, good cooks. I think God makes good cooks to make good food. And, you know, if the food happens to be made in the lab, great, I think, but I don't know, discuss. Other people might have different views, but
a fun
yeah,
note to end on. So thank you very much for all your input on bioethics. This is